This is a quick overview of the ca43k app at shinyapps.io. This is a bioconductor-based application that helps us search through 43,000 RNA-seq studies related to cancer. It has an About tab here, which tells us some of the details of the underlying techniques for constructing this app. And there's a collection of titles for the studies that we've selected through this selector, which has an index to keywords of all of the sample attributes and other metadata about these studies. Each of the tabs gives the sample level metadata about each one of these studies. And we can construct a RESTful summarized experiment by clicking on the rows of the studies, the rows of this title, to acquire information about the studies that we're interested in. If we take the Download Summarized Experiment button, this will set up distribution to my disk of a certain summarized experiment. I'll save that, and let's have a look at what we got. We'll go into RStudio, and let's see, get WD. We will do a mySE. Let's move this up a little bit. Equals read RDS of SE3. Oh, maybe it's 31. What did we get there? 20. 0331.rds. And this is a summarized experiment instance. It has 314 samples. That would be the sum of 20 plus 9 plus 290. Perhaps some of the samples listed in these tables are not available, but the total number in our RStudio experiment here is 314. And the metadata about each one of these studies is available in the metadata component here. Metadata mySE 2, for example, is this data frame about the 20 samples. And one can see that the genotypes of the individuals in this study could be wild type or mutant for BRAF. If we look at the other study here, here we have only 100 rows, but we can see that there's a great many different types of tumor in this study. And the reason that this study got in is because some of these tumors had reference to BRAF mutations. So this app is available at shinyapps.io. It makes use of the package called Human Transcriptome Compendium, where the general data structure is defined, and it also makes use of a package called SSRCH, which takes care of indexing heterogeneous metadata. Let's take one last look at the result here. If we do an assay of mySE, we see that this is a delayed matrix because the actual quantifications are not present on your machine, but they are present in the HDF Scalable Data Service instance in the Amazon Web Services buckets. And that has been provided very kindly by John Reedy of the HDF group. So this is a very quick overview of this app, And if you have questions, please don't hesitate to file issues at the GitHub repository for the Human Transcriptome Compendium, which is at VJCITN in GitHub. We'll take a quick look at that. There. Thank you for your time.